Good morning. Clearly, all the important people are here, and those who. But I think we can make a start. It's my very great pleasure and privilege this morning to introduce Angela Rivallese, who will really be known to everybody here. She's a full professor of food and dietetic science at the Federico II University of Naples. She has made a huge contribution to diabetes in general, lipid metabolism, metabolic disease, and cardiovascular disease through a large number of clinical trials which she has conducted over the years, and has made a contribution in many ways, not least of all as one of the chairs of the board of the Diabetes and Nutrition Study Group. I think it's very appropriate that Angela be asked to give one of the key plenary lectures at this conference. And Angela, we really do look forward to hearing some of your more recent work.
Is it working? Yeah. Thank you very much, Jim, for your kind presentation, but you are a friend, so it's not good. Thank you. I would like to start thanking Ulf Riserus, all the organizers of this symposium, for giving me the opportunity to give this lecture. My presentation will focus on the possible role of the liver and the colon in the relationship between nutrition and metabolic disease. That is an emerging topic of the literature and is one of the topics of the research of our group. Very strong and consistent evidence indicates very clearly that dietary habits may influence the progression, the onset and the progression of a metabolic disease, in particular type 2 diabetes, and more in general, the health of human beings. Among the different evidence present in the literature, I choose to show you the data of this meta-analysis carried out by the study group, showing that the intervention on the lifestyles, physical activity, but especially type of diet in the right direction, is able to halve the risk of type 2 diabetes. This very strong relationship may be explained by the fact that everything we introduce with the diet, after the process of digestion and absorption, enters into intermediate metabolism and then arrives to cells, regulating their structure and their function. All the organs are very important in this interplay, but certainly a fundamental role may be played by the liver, which is the first organ where the product of digestion and absorption arrives, and is also a fundamental organ in regulating the intermediate metabolism, glucose metabolism, lipid metabolism, proteic metabolism, and so on. Moreover, another organ very important in this interplay is certainly the intestine. In the past years, much attention was paid to what happened in the small intestine, at the level of the stomach, of the small intestine, for what concerns the process of digestion and the absorption. And this process still remains very important. But in recent years, more attention has been given to what happens in the colon. But we will come back to this later on. Let's start with liver. Certainly, diet, principally for the high energetic intake, but also for its composition, affects very much the accumulation of fat at the liver level, leading so to the hepatic steatosis, which is the first step of non-alcoholic fatty liver disease, which is now the most frequent liver disease and is very frequent, especially in obesity and type 2 diabetes, and may be implicated also in the pathogenesis of this metabolic disease, since liver fat is strictly linked to insulin resistance, especially at the liver level. The positive energy balance and is very important in affecting liver fat. The first step for the prevention and the treatment of non-alcoholic liver disease is certainly the weight reduction. And in fact, there are a certain number of intervention studies showing that a reduction of 7–10% of weight is able to reduce liver fat from around 30% to more than 50%, which is not only significant, but is only clinical relevant. And this effect is independent on the composition of the diet. These results have been recently confirmed also in the long run after 1 year in the direct study, where a more sustained weight loss of about 10–15% was able to completely normalize liver fat with a subsequent improvement in insulin resistance, especially at the level of liver. But as I said before, also the composition of the diet may influence the liver fat. And at the beginning, the attention was focused on the possibility of the effects of different types of diet. And more than 10 years ago, Lutgarda Bozzetto of our group showed that a diet rich in monounsaturated fat, deriving mainly from extra virgin olive oil, was able to reduce in type 2 diabetic patients liver fat by 27%. And in the same period, the group of Rivallese showed that similar results in high cardiometabolic risk individuals, substituting saturated fat with omega-6 polyunsaturated fat. At the same time, data from the literature, based especially on epidemiological studies, but also on some intervention studies, really with non-univocal results, show that other components of the diet, such as whole grains, dietary fibers, legumes, low fructose intake, omega-3 PUFA, polyphenols, and some vitamins, may be associated with a better outcome in terms of liver fat. So our question was, when can we achieve better results with a multifactorial diet resembling a Mediterranean diet, but containing also other components such as polyphenols, typical of other healthy dietary patterns? So we tried to answer these questions, evaluating the effects of this kind of diet, the multifactorial diet, compared with a MUFA diet, rich in monounsaturated diet, that this was the diet already proven to be able to reduce liver fat. The main foods characterizing the two diets are shown in this slide, and for what concerns multifactorial diet, the main foods are whole grain cereals, pulses, some vegetables, almonds, some kinds of fruit, fish, extra virgin olive oil, green tea, and decaffeinated coffee. The main foods were given to participants, so the compliance was optimal. And the two diets are essentially different concerning the content of omega-3 polyunsaturated fatty acid, polyphenols, and especially fiber. Instead, the composition, the nutrients, the percentage of nutrients, carbohydrates, fat, and protein was similar between the two diets. After 2 months, the multifactorial diet was able to reduce liver fat by 40%. And this reduction was higher compared with the reduction obtained with the other diet. And was very similar to that obtained with hypocaloric diet, so with weight loss, and also very similar to the results obtained with the extreme diet, such as a ketogenic diet, and both of these diets are very difficult to be followed for a very long period of time. But coming back to the possible role of the liver, we have to analyze the possible mechanism, especially at the liver level, that can explain these results. The deposition of fat at the liver level is the results of the net balance between metabolic pathways favoring the uptake and storage of lipids and the metabolic pathways favoring instead the disposal of lipids. Among the metabolic pathways favoring the uptake and storage, there are the uptake of fatty acid from the diet, but especially the uptake and storage of fatty acid coming from adipose tissue. And this pathway is very important, very relevant when the ability of adipose tissue to storage lipids is overcome. And the storage depends also on the de novo lipogenesis, the lipogenesis of fat, of endogenous fat derived from non-lipid precursors. Dietary intervention may act on different steps of this complex mechanism. And in particular, for example, weight reduction perhaps may act especially on the reduction of fatty acid coming from adipose tissue. In our isoenergetic intervention studies, we tried to evaluate some of these steps, and in particular, we evaluated the levels of beta-hydroxybutyrate, which is considered a good index of the fat oxidation, and the ratio between palmitic and linoleic acid in serum triglyceride, which is an index of de novo lipogenesis. With the first experiment with the diet rich in monounsaturated fatty acid, there was a significant increase in fat oxidation, especially in the postprandial period. And this increase in fat oxidation were related to the reduction of liver fat. Instead, with the multifactorial diet, I remember that this diet has the same amount of monounsaturated fatty acid of the other diet. Instead, we showed we had a reduction, significant reduction of the novel lipogenesis index compared with the other diet. And this reduction was about 30%. And moreover, the reduction of lipogenic index was correlated with the reduction of liver fat, suggesting a possible role of the reduction of de novo lipogenesis in the decrease of liver fat. But liver fat is generally associated with the deposition of fat in other noble tissue, such as heart, but also pancreas, and the deposition of fat into pancreas may impair either the exocrine function or the endocrine function of the pancreas. And in particular, it has been demonstrated on the basis of animal studies and in vitro studies that the deposition of fat in the pancreas may impair the insulin secretion through different mechanisms. And it is to note that our multifactorial diet was able to reduce also pancreatic fat compared with the other diet. The reduction was about 10%, almost similar to that obtained with a very sustained weight loss in the direct study. And this decrease was accompanied by an increase, not change in glucose response to a test built, but was associated with an increase in the insulin response to a test meal, especially in the first 2 hours. So in the early response to insulin resistance, which is considered an index of a better function of beta cells. And this reduction was inversely related with the increase. The reduction of pancreatic fat was inversely related with the increase of the insulin response in the first 2 hours after a test meal, suggesting a possible role of the decrease of pancreatic fat in the improvement in the insulin secretion, especially in the postprandial phase and especially in the first part of the postprandial phase. And it's important also to realize that there was no relationship between the decrease in pancreatic fat, the decrease in liver fat, suggesting a possible different mechanism, such as some data suggest also from the literature. And the decrease in pancreatic fat was not related to decrease in de novo lipogenesis, as it was liver fat, but the only factor related to the reduction of pancreatic fat was the reduction that we found in the plasma levels of ApoC3, that is an inhibitor of lipoprotein lipase. But of this result will be shown to you later on by Pina Costabile of our group. Whatever the mechanism, anyway, our results are in line with the results obtained in the direct study, where a sustained weight loss was able to normalize liver fat, to reduce pancreas fat, but was able to induce remission of diabetes only in people able to restore their beta cell function. Of course, we cannot speak in our study about remission for the very short duration of the study, but I think that our data suggest that the twin cycle hypothesis, giving a great importance to the liver and to the fat in the pathogenesis of type 2 diabetes, may be worked also in an isocaloric condition. And in this condition may be modulated by the composition of the diet. And I think that it is very important from a clinical point of view for the identification of the best composition of the maintenance diet. Summarizing the first part of my presentation, I think that we can say that diet, both from the point of view of energy intake and its composition, significantly influences hepatic steatosis. The mechanism of action are different depending on the type of nutrition intervention, and many of them occur at the level of liver. Diet also influences other fat in pancreatic fat likely with a different mechanism, and these mechanisms are until now less identified. The reduction of pancreatic fat is associated with an improvement in the early insulin response. Therefore, nutrition influences both the insulin resistance and the beta cell function, two of the factors leading to type 2 diabetes. Let's move to the other part, to the possible role of the colon. The product of the digestion and the product of foods not absorbed in the small intestine. Here in this slide, I'm referring to resistant starch, to fibers and so on, but this works also for the other components of the diet. This product reach the colon, and in the colon, they can modify the profile of intestinal microflora. And before this, then the modification of intestinal microflora may act on different steps, may act on subclinical inflammation, or the integrity of intestinal barrier, but also on the fermentative capacity of intestinal microflora with the production of the short-chain fatty acid. And this short-chain fatty acid, after absorption, may influence in a positive way, depending on the type of short-chain fatty acid, glucose metabolism, lipid metabolism at the liver level, but also at the level of the other organs, may act also on the degree of satiety, acting also at the brain levels, and may act also on other different metabolic functions. The production of short-chain fatty acid is strictly linked to fiber and to non-digestible carbohydrates. And I will focus on this aspect, especially on the effects that different types of diet may have on the production and the plasma levels of short-chain fatty acid. And in particular, with a particular attention to propionate and butyrate, which are the two short-chain fatty acids for which animal studies, also in vitro studies, show that indicates that the possible beneficial effect on glucose metabolism may be due to different mechanisms, but especially on improvement of the insulin sensitivity or reduction of insulin resistance, especially at the liver level. Some years ago, Rosalba Giacco of our group showed that a diet rich in whole grain cereals compared with a diet rich in refined cereals was able to reduce, to improve postprandial insulin action, and was able to reduce the levels of postprandial triglyceride. And in this study, as Gabriele showed you yesterday, there was an increase in propionate, in fasting propionate, which correlated with the intake of cereal fiber, and the increase in propionate was associated with reduced levels of insulin at in the postprandial phase, suggesting a possible role of propionate in the improvement in insulin resistance. But all we know, and Gabriele Riccardi showed you yesterday, the effects of fiber on the fermentative capacity of intestinal microflora may depend very much by the different types of fiber. So what happens with a diet rich in natural foods, rich essentially in different types of fiber, so soluble, insoluble, deriving from different natural foods. And we tried to give an answer to these questions with a new intervention study in non-diabetic individuals, but in individuals with a high cardiometabolic risk, evaluating the effect in this case of a real typical Mediterranean diet, so rich in whole grains, fruit, vegetables, dried fruits, legumes, extra virgin olive oil, fish, with rich in different types of fiber, both cereal and non-cereal fiber, and low instead in red meat, cheese, and so on. Comparing the effect of this diet with a control Western diet. And the aim of this study was to evaluate the effects of these two diets on glucose metabolism, especially in the postprandial phase, and on the intestinal microflora. After 2 months of the multifactorial diet, there was a significant reduction in blood glucose levels at the end of the study after a test meal compared with the other diet, but even more, a significant reduction in insulin in the postprandial phase with an improvement in insulin sensitivity in this phase, in this postprandial phase, evaluated by OGIS. And what is more important, with the Mediterranean diet, there was a change in microflora, especially an increase in the abundance of Akkermansia mucifera, and especially the change was quite relevant for what concerns species able to degrade fiber, such as Roseburia and Faecalibacterium prausnitzii. And in this case, we didn't find any change in propionate, but instead there was a significant increase in the butyrate, but only in the postprandial period. And this increase in the butyrate was related to improvement in insulin resistance. So I think that with our intervention, we have showed that nutritional intervention, in this case a Mediterranean diet, was able to improve some metabolic pathway. On the other hand, this nutritional intervention was able to change the microbiome, especially with an increase in fiber able to degrade fiber, and with an increase in postprandial butyrate, which in turn may have induced the metabolic improvement. Our data are quite in line with some data present in the literature. In fact, 2 of the bacteria increased in our intervention, Akkermansia and the Faecalibacterium, are among the bacteria that in this review of association studies are inversely related both with the plasma glucose levels and also with the plasma insulin levels. Before concluding, our group is now interested also in evaluating the relationship between diet, microflora, and blood glucose control in type 1 diabetic patients. That is another very important topic of the literature, and for which there are very few studies. Preliminary data show a very interesting relationship between the adherence to Mediterranean diet, the change in the profile of microflora, and the blood glucose control. But these results will be shown to you by Jumana later on in the next session. So I think that we can conclude that nutrition and metabolic disease are closely connected. The liver colon play a fundamental role in this complex relationship, both as a target organ, but also as main players regulating the insulin resistance and the beta cell function, two of the most important factors leading to type 2 diabetes. And for, I don't know if you can see anything, but I would like to thank the members of our group who participated in these studies. I can say this is the historical group. You see me, Gabriele, Rosalba Jack, and so on. We participated since the beginning to the life of this study group. Here is the youngest of our group, which represent the present and the future of our group. And many of them have participated actively also to this symposium. And I would like also to thank the study group. I found some photo of the old symposium of this study group, starting from the beginning, where Monica, Jim, and Gabriele were very implicated in drafting. I don't remember if the first rules of our study group or the first guidelines of our study group. And also found a photo of Brita, of a people for Japan, who for many years participated to our symposium, and Jenkins and Riven debating always since the beginning, and also Islam. I can say that we grew up together with this study group, and I hope, I am sure that also the youngest of our group will continue to be a very active part of this study group, and perhaps they will organize a next symposium in Naples or near Naples. Thank you for your attention.
Angela, I would like to thank you on behalf of everybody for a wonderful talk which you gave us. It was a brilliant plenary lecture. There was to have been something else happen. There was to be something else happening right now. I was asked by the board to present you with a certificate of the honorary membership of the group because of the wonderful work that you've done, both in terms of your science, but also in terms of what you have done for the group. There's something gone wrong with the printing mechanism, I understand. And you will be presented with that later. But on behalf of all of us, congratulations on that too. And thank you.